Bladder EpiCheck real-time PCR with rotor gene Q instrument platforms. Materials required but not provided. Strip tubes and caps, 0.1 milliliters, 72 well rotor loading block, filtered tips, 1.5 milliliter tubes or strip tubes, mini centrifuge. Use the test form for reaction mix preparation. Calculate the reaction mix for patient samples, 2 controls, and 1 extra for dead volume. Completely thaw the following reagents at room temperature: digested DNA samples, if they are frozen, PCR mixes A through H, Taq DNA polymerase. Cover qPCR tubes while thawing as they contain fluorescent dyes that are light sensitive. Once thawed, mix each tube before use. Do not vortex. Briefly spin down to remove drops from inside the lid. For PCR mixes preparation, you will need 8 clean tubes labeled A through H. Prepare assay mixes according to the table in the protocol and in the test form. Load qPCR mixes A through H according to the number of samples. Load Taq polymerase according to the number of samples. Mix each tube. Do not vortex. Mix digested DNA samples and controls as well. Briefly spin down to remove drops from inside the lid. Place the 0.1 milliliter PCR strip tubes in the 72 well loading block according to the number of patient samples, including UC and NTC controls. PCR mixes are loaded by rows. Pipette 15 microliters of assay mixes to the corresponding wells. Assay mix A into the first row, assay mix B into the second row, etc. The number of columns correspond to the number of patient samples and controls. In order to avoid cross-contamination, press the pipette plunger into its first stopping point only. Change tips between mixes to avoid cross-contamination. Use a full box of pipette tips to load digested samples and controls to allow visual tracking. Samples are loaded by columns. Pipette 10 microliters of each digested DNA sample and control according to columns. Full column per sample. Load NTC exactly to positions number 1 through number 8, and UC exactly to positions number 9 through number 16. Change tips between wells. Do not discard the sample residuals. They could be further diluted and used for PCR repeats. Keep them at -20 degrees Celsius. Gently tap the rack to allow the samples to reach the bottom of the tube. Close the strip tubes using 0.1 milliliter strip caps and mark the first cap for correct positioning inside the rotor. Always run a full rotor. In case less than 7 patient samples are analyzed, fill the empty positions with tubes filled with water. Do not use tubes with PCR product to balance the rotor. Place strip tubes on the 72-well rotor to match the tube numbers to the numbers written on the rotor. Attach the locking ring. Points to remember. Store digested DNA samples at -20 degrees Celsius for up to 30 days. In order to minimize cross-contaminations, separate areas, racks, and pipettes for template, non-template, and PCR mix setup. Each run analysis must include digested UC and NTC samples. Do not vortex reagents. Avoid exposure of PCR reagents to direct light and/or extreme temperatures for extended periods of time. Load NTC exactly to positions number 1 through number 8 and UC exactly to positions number 9 through number 16. Always run a full rotor. In case of empty positions, load tubes with water. Proceed to PCR instrument setup run analysis according to the instructions for use.